Bismillahirrahmanirrahim. Mechanism of enzyme action. How enzymes work have been proposed by Macallis and Manton. They proposed a hypothesis for enzyme action which is most acceptable at that era. According to their hypothesis, the enzyme molecule first combined with substrate to form an enzyme-substrate complex, which further dissociates to form a product and free enzyme. Here, enzyme once dissociated from the complex is free to combine with another molecule of substrate and form a product in a similar way. The enzyme substrate complex is an intermediate or transition complex, and bonds involved are weak non-covalent bonds such as hydrogen bonds, van der Waals forces, and hydrophobic interactions, etc. Now, the models of enzyme substrate complex formation. These interactions have been described basically of two types. Number one, template or lock and key model, and number two, induced fit model or Koshland model. Number one, template or lock and key model proposed by Fisher, which states that the active site already exists in a proper conformation even in the absence of enzymes. Thus, the active site by itself provides a rigid pre-shaped template fitting with the size and shape of the molecule, with the size and shape of the substrate molecule. Here the substrate fits into the active site of the enzyme as the key fits into the lock. This model proposes the substrate binds with the rigid pre-existing template of the active site, provides an additional group for binding with other ligands. This model cannot explain the change in enzymatic activity in the presence of allosteric modulators or inhibitors. Induced fit or Koshland model. Induced fit or Koshland model proposed by Koshland in 1963 is known as induced fit model. The important feature of this model is the flexibility of the region of the active site. According to this, the active site does not possess a rigid preformed structure as the enzyme to fix the substrate. According to his hypothesis, the substrate during its binding induces the conformational changes in the active site to attain the final catalytic shape and forms. This model explains the several matters related to the enzyme action. That includes the enzyme model become inactive on denaturation. It able to explain the kinetic saturation. It able to explain the competitive and non-competitive inhibitions, their mechanisms, as well as it able to explain the allosteric modulations. Mechanism of enzyme action can be viewed from two different perspectives. First, treats the catalyst in terms of energy changes that occurs during the reaction. That is the energy the enzymes provide and alternate energetically favorable reactions pathway that differs from the uncatalyzed reaction. And the second perspective describes that how the active sites chemically facilitates catalyst. Energy changes occurs during the chemical reactions. As we know that all chemical reactions have an energy barrier that separates the reactants and product. This barrier, called the free energy of activation, is the energy difference between that of the reactants and the high energy intermediates that occurs during the formation of products. Look at the figure. Figure shows the change in energy during the conversion of a molecule of reactants A to a product B as it proceeds through the transition state, the high energy intermediate. Free energy of activation. The peak of energy is the difference in the free energy between the reactants and the product where the high energy intermediate is formed. It means where the maximum enzyme substrate complex is formed during the conversion of reactants to a product. Because of high free energy of activation, the rate of uncatalyzed reactions are often slow. Rate of reactions for a molecules to react should be should contain sufficient energy to cross the energy barrier from reactants to a product after achieving a transition state. In the absence of enzyme, only a small fraction or proportion of a molecules may possess enough energy to achieve the transition state between the reactants and product. Rate of reaction is determined by a number of such energized molecules. In general, lower the free energy of activation. In lower the free energy of activation, the more movement of maximum molecules have a sufficient energy to cross the barrier from reactant to product by achieving the transition state represented by T star. Alternate reaction pathway. An enzyme allows a reaction to proceed rapidly under conditions that prevailing in the cell by providing an alternate reaction pathway with a decreased free energy of activation. Enzymes does not change the free energies of reactants and products, therefore does not change the equilibrium of the reaction. It does not, however, accelerate the rate with which the equilibrium is established. Now the chemistry of active site. The active site is a complex molecular machines employing a diversity of chemical mechanisms to facilitate the conversion of substrates to products. A number of factors are responsible for the catalytic efficiency of enzymes, including the transition state stabilization and some other mechanisms. In transition state stabilization, the active site often acts as a flexible molecular template that binds the substrates and initiates its conversion to the transition state. That is a structure in which the bonds are not like those which exist in the substrate or the product. By stabilizing the transition state T star, the enzymes greatly increase the concentration of reactive intermediate, that is, in transition state stabilization. Enzymes able to convert a maximum amount of substrate into a enzyme substrate complex that can be converted into a product and thus accelerate the rate of the reaction. Other mechanisms. Active sites can provide a catalytic group that enhance the probability that increase the transition state and thus the transition state is formed. In some enzymes, these groups can participate in general acid-base catalysis in which the amino acid residues provide and provide or accept the protons. Visualization of transition state. The enzyme catalyzes the conversion of substrate to product can be visualized. The substrate binds to the enzyme during the enzyme substrate complex formation is at a lower energy than unbounded substrate and explain the small dip in the curve at the enzyme substrate complex. Recommended books Lippincott 55 and 56 page number, Chatterjee 124, 8th edition. Assignment enumerate all mechanisms through which the enzymes cause catalyst. B. What is the mechanism of enzyme catalyst? Graphical representation.